everyone. This is the sixth lecture for your chapter Photosynthesis. In the last class, we discussed Calvin cycle, which is the main, cycle of your biosynthetic phase, okay, or dark reaction. Fine, we had seen light reaction, then we moved on to dark reaction, which is a misnomer, okay, and it is better known as biochemical phase or biosynthetic phase. Clear? In that, we studied the Calvin cycle. Just a quick overview, a quick summary. We start with ribulose 1,5-bisphosphate, which is the first carbon acceptor molecule. Okay, it is a 5-carbon molecule. It will accept one carbon dioxide. Okay, and this carbon dioxide will be added to it. The step is called carboxylation, and the enzyme is called Rubisco. Right, ribulose bisphosphate carboxylase oxygenase. Fine. After that carboxylation occurs, it forms the first stable product of your Calvin cycle, which is nothing but 3-phosphoglyceric acid. Okay, 3-PGA, phosphoglyceric acid. 3 is the position where the phosphate is, C. Right. Now this 3-phosphoglyceric acid will be reduced. So the second step is reduction. In this step, in the reduction step, 3-phosphoglycerate is first converted into 1,3-bisphosphoglycerate, and that 1,3-bisphosphoglycerate is finally converted into 3-phosphoglyceraldehyde. Fine. So from glycerate, which is carboxylic acid, it is reduced into aldehyde. Okay, so 3-phosphoglyceraldehyde. In this reduction step, you require 1 ATP and 1 NADPH per molecule of 3-phosphoglycerate. Okay. After reduction, you have now 3-phosphoglyceraldehyde, PGAL. So, two molecules of PGAL will go and make sucrose, starch, glucose, fructose, all of the sugars. Fine. First of all, of course, it will make glucose. That some of the glucose will be converted into fructose. Then glucose plus fructose, it will make a disaccharide, okay, which is sucrose. This sucrose is the transport sugar. It will be transported through the vascular bundle, and finally, wherever it has to reach the sink, right? It reaches the sink, the tissue where it is required. At that place, sucrose is again divided into glucose plus fructose. That glucose is used to make starch, fine, which is the storage polysaccharide. Fine. So this was the whole step after reduction. Now the third step is called regeneration. 2 molecules of phosphoglyceraldehyde will go for synthesis of sugar, but 10 molecules of phosphoglyceraldehyde will go and make 6 molecules of ribulose phosphate. Now, at the starting, we have ribulose bisphosphate. So, this ribulose phosphate molecules are phosphorylated, okay, using ATP. So, you get 6 molecules of ribulose 1,5-bisphosphate back. Fine. So, this is the regeneration step. It requires one more ATP. Fine. So, this is what the Calvin cycle is. Okay, it is the basic most cycle of photosynthesis, and it is conserved. Okay, it is the oldest cycle and it is conserved in all different species. Now remember, we had discussed that scientists were curious, does every plant have first stable molecule as phosphoglycerate? So the answer is no. If you remember, we had also seen that the first stable molecule can be oxaloacetate, which is a 4-carbon compound. So the plants that are adapted to dry and tropical regions, okay, they are adapted to dry and tropical meaning the humidity may also be high. If not, then at least the temperatures are higher comparatively. Fine. So dry tropical regions, they have C4 pathway. It is comparatively recent pathway. Okay, it has evolved after C3 pathway. Here what the plants do is they are called C4 plants. First thing, so C4 plants have oxaloacetic acid as their first stable carbon fixation product. We had seen carbon fixation, carbon assimilation, all of these mean the same thing. Okay, basically your carbon dioxide from atmosphere is getting added to some organic compound. Fine. So that is your carbon fixation, and the first stable product is oxaloacetate. Calvin cycle, once again I would like to remind, is still the main biosynthetic pathway. The C4 plants have a special type of leaf anatomy for this whole pathway. Fine. Here you can see the basic C4 pathway is what? You have carbon dioxide, which will form a bond with PEP, phosphoenol pyruvate. If you remember phosphoenol pyruvate, it comes in glycolysis as well. Okay, it is a 3-carbon compound. It forms a bond with carbon dioxide. The enzyme is called PEP carboxylase. Okay, so it is phosphoenol pyruvate carboxylase. Carboxylase, the enzyme does what? It does the addition of carbon dioxide, and you get the first 4-carbon compound, which is oxaloacetate. Let us see what this special structure is, okay, in C4 plants. So in C4 plants, this special structure is called Kranz anatomy. Kranz means this wreath. Have you seen such kind of flower wreath? They normally, you must have seen the prime minister and president and all putting such sort of wreath on different memorials, right? Also in Hollywood movies, you may have seen that people put such sort of wreath on their doors, right? So this here is called a wreath. Now, when you look at a C4 plant's leaf, it looks like this. Okay, can you see the resemblance? It looks like a wreath. Fine. So this is called Kranz anatomy. Clear? Now, in Kranz anatomy, what is this Kranz anatomy? So remember mesophyll cells. Mesophyll cells are the cells present in your leaf that have chloroplast and do the function of photosynthesis. So here the outermost layer of this columnar like cells is called your is your mesophyll cells. Clear? Inside your mesophyll cell there is another layer. Can you see? This another layer of cells is called bundle sheath cells, and uh, inside this bundle sheath cells is your vascular bundles, okay, your xylem and phloem. So this is how the concentric circles, okay, they are arranged. You have outermost mesophyll cell, then you have bundle sheath cell and innermost vascular bundle. If you see here, this is a microscopic diagram, okay. When you take a cross section of leaf and look at it under microscope, it looks like this. One more thing I would like to say is the mesophyll cells here, okay, they do not have many chloroplast, okay. Very rarely you find mesophyll cells having chloroplast. So C4 plants specialty is it does not, the mesophyll cells of C4 plants do not have chloroplast. While when you look at the bundle sheath cells, which are these inner spherical cells, they have a lot of chloroplast. Hence you can see they are dark green in color, right? And in between is your vascular bundle. So if you go from inside to outside, you have xylem, phloem, bundle sheath cell and mesophyll cell, all of which are connected through plasmodesmata. Clear this much? Okay. So now let us study this C4 pathway. What is this C4 pathway, or which is also known as Hatch-Slack pathway? In C4 pathway, the primary carbon dioxide acceptor is a 3-carbon molecule called phosphoenol pyruvate, PEP. And this PEP, okay, phosphoenol pyruvate, is found in the mesophyll cells. Okay, so the outermost cells, the mesophyll cells, they have phosphoenol pyruvate. So what is the whole reaction? Carbon dioxide plus PEP, which is a 3-carbon compound, will give you oxaloacetate plus which is a 4-carbon compound. Okay, so carbon dioxide plus PEP giving you oxaloacetate. The enzyme here is PEP carboxylase, okay, or PEPcase it is known as in short, which is nothing but PEP, okay, phosphoenol pyruvate carboxylase. In C4 plants, the mesophyll cells, they lack Rubisco enzyme. So here the first enzyme is not Rubisco, pay attention, and the mesophyll cells themselves do not have Rubisco enzyme. One thing I had forgotten to mention was, um, I don't know if anybody noticed, but when you write the full form of this enzyme, which is ribulose bisphosphate carboxylase oxygenase. So it is also known as oxygenase because this enzyme, okay, Rubisco, it was actually formed when the atmosphere of Earth was still reducing type. Okay, we shall study that in evolution. So in evolution, if you remember, you must have studied different phases of um, uh, evolution of life and how the Earth was primitive Earth. So when the life it originated, okay, when the first photosynthetic cells originated, at that time the atmosphere was reducing. There was not much oxygen which was present in the atmosphere. So this enzyme Rubisco developed at that time. Later on, what happened was the enzyme remained the same, but now there is more oxygen in the atmosphere compared to carbon dioxide. So it is oxidizing environment. This enzyme has an active site which can also bind to oxygen. Okay, so to prevent that, Rubisco enzyme here is missing in the mesophyll cell. You will understand all of this when we study photorespiration. Okay, we will study photorespiration when it will all become clear to you. Right. So ribulose bisphosphate carboxylase oxygenase. Okay, it can add oxygen also to ribulose bisphosphate. It can also add carbon dioxide. Hence the name. Okay, so let us study this pathway using this diagram. The first, the outermost cell is the mesophyll cell. Then you have bundle sheath cell, and this is your vascular tissue. Carbon dioxide will enter the mesophyll cell, okay, and inside the mesophyll cell there is presence of pepcase or pep carboxylase. This pep carboxylase will convert carbon dioxide and PEP into 4 carbon compound oxaloacetate. Oxaloacetate will get converted into another enzyme called another organic acid called malate or malic acid. Malate again is a 4 carbon compound. We shall study malate and oxaloacetate again in Krebs cycle. Okay, from malate in Krebs cycle you are making oxaloacetate from malate, and here from oxaloacetate you are making malate. Clear? So this is the reverse reaction, oxaloacetate to malate. This malate can now enter the bundle sheath cells. Okay, so it will enter the bundle sheath cell. As soon as it enters, it will get divided into two parts. Malate, which is 4 carbon, will release carbon dioxide and get converted into pyruvate, which is a 3 carbon compound. So now you have this carbon dioxide and pyruvate. Pyruvate, which is 3 carbon compound, will again be converted into phosphoenol pyruvate. So this is the regeneration step. Okay, and this PEP will now be present in the mesophyll cell. Okay, this pyruvate will move out into the mesophyll cell and get converted into PEP. This process, these steps require ATP. But when you look at the phosphor what you can say the ATP, okay, that ATP is getting converted into AMP. Okay, ATP is getting converted into AMP, which is equivalent of using 2 ATP. Fine, so here you can either write 1 ATP to AMP, or you can write 2 ATP are required for this whole process. Clear? 2 phosphates are utilized. Hence, you can say 2 ATP are used. Now this carbon dioxide is present in the bundle sheath cell, right? So you have basically separated the cell which will be accepting all the gaseous molecules from the cell which will actually be doing the Calvin cycle. Clear? So what will happen in this kind of cell, that more and more carbon dioxide will be fixed and it will be released here in the bundle sheath cell. Clear? Fine. So this carbon dioxide, it will enter the Calvin cycle. Okay, there will be Rubisco present here also. The whole same cycle will run and you will get sugar. This sugar will enter the vascular tissue and it will be allowed to go wherever it is required. Right. So in mesophyll cell, carbon dioxide plus PEP gives you oxaloacetate. Oxaloacetate gives you malate. In bundle sheath cell, the oxaloacetate will give you carbon dioxide plus pyruvate, and this this pyruvate again, it will go back into the mesophyll cell. It will use one ATP, okay, and get converted into AMP. Here I have made a mistake. It is AMP, okay, so which is equivalent of using 2 ATP. This PEP is again ready for the next cycle. The carbon dioxide released in bundle sheath cell enters the Calvin cycle, as we have already seen, and this is the diagram that was given. Okay, so here you have the mesophyll cell and here is your bundle sheath cell. The first thing that is happening, carbon dioxide plus phosphoenol pyruvate, there is fixation, okay, which will give you C4 acid. Here oxaloacetic acid and malic acid, both of them are organic acid having 4 compounds. So you can just write it as C4 acid. It is fine. C4 acid which will enter into the bundle sheath cell. So there is transport. Then C4 acid will be decarboxylated. Decarboxylated meaning the carbon dioxide will be removed. So here you can see the carbon dioxide being released, and C4 acid gets converted into C3 acid, which is nothing but pyruvate. This C3 acid is transported back into the mesophyll cell, fine, and this transport after that it gets converted into phosphoenol pyruvate, and this process is called regeneration. Okay, so fixation, transport, decarboxylation, again transport, and regeneration. Clear? Let us come to the differences between C3 and C4. It has been a while since we have seen differences, right? Okay, so C3 pathway, here the carbon dioxide is fixed only once, only by Rubisco, and in the mesophyll cells, what will happen is carbon dioxide plus RUBP will give you 2 molecules of 3-phosphoglycerate. So it is fixed only once. While in case of C4 pathway, carbon dioxide is fixed twice. As you can remember, you have PEP carboxylase, which will fix the carbon dioxide from atmosphere and make oxaloacetate. And second time, the Rubisco enzyme in the bundle sheath cell will convert that carbon dioxide into RUBP. It will convert that carbon dioxide plus RUBP into 3-phosphoglycerate. Okay, so the first acceptor here is RUBP in case of C3, while the first acceptor in case of C4 pathway is phosphoenol pyruvate. The second acceptor of carbon dioxide is RUBP. Carbon dioxide fixing enzyme, the first enzyme is Rubisco. Okay, the only enzyme in fact in C3 pathway is Rubisco, while in case of C4 pathway, the first enzyme is pepcase, and the second enzyme, when the second time the carbon dioxide gets fixed, it is Rubisco. Cranzenatome is absent. In the C4 pathway, it is present. To fix one carbon dioxide molecule, you require 3 ATP plus 2 NADPH. Remember, we had already studied this. Okay, for one carbon dioxide, will give you 2 molecules of 3-phosphoglycerate, and for 2 molecules of 3-phosphoglycerate during reduction, you require 2 molecules of ATP plus 2 molecules of NADPH. Why it is written 3 here is because during regeneration, okay, when you are going to make RUBP again, you require one more ATP. So for one carbon dioxide, you need 3 ATP plus 2 NADPH. To fix one carbon dioxide in case of C4 pathway, you require 5 ATP plus 2 NADPH. Okay, 3 ATP plus 2 NADPH remains the same. During Calvin cycle, you require the same amount. Why the number of ATP is higher here is because from pyruvate, when you want to regenerate PEP, you require 2 ATP. Okay, so 2 plus 3 gives you 5 ATP plus 2 NADPH. So for one glucose molecule, this was for one carbon dioxide. If we want to talk about one glucose, which is everything multiplied by 6, right? 6 carbon dioxide make one glucose. So for one glucose molecule, you require 18 ATPs and 12 NADPH in case of C3 pathway. while in case of C4 pathway you require 30 ATP, okay, 5 6 are 30 plus 12 NADPH. The C3 pathway requires higher carbon dioxide concentration. It is shown in brackets, which means I'm talking about the concentration of carbon dioxide, and it is higher. It should be higher in the atmosphere for C3 pathway to work efficiently. While in C4 pathway, it can also function at low atmospheric carbon dioxide concentration. Why? Because whatever carbon dioxide it will be able to catch, it will transfer it to the bundle sheath cells, and it will get concentrated in the bundle sheath cell where exactly the Calvin cycle is taking place. So Calvin cycle will never run out of carbon dioxide. Almost 95% of the green plants are C3 plants because it is the oldest cycle, okay, Calvin cycle, it is the oldest cycle, and C4 pathway is a relatively new evolved pathway. So about 5% of the green plants are C4 type of plants. Now let us compare the examples of C3 pathway plants or C3 plants. They include rice, wheat, oat, barley, cotton, peanuts, tobacco, sugar beets, soya bean, and spinach. So as you can see, it is mix of both. It has monocots as well as dicot plants. Fine. While in case of C4 pathway, it is maize, sugarcane, pearl millet is bajra, and sorghum. Fine. So these mostly you will see are monocot plants. Okay, more monocots, less dicots. While in this case, C3 plants you can find both monocots as well as dicots. Fine. So just remember these examples also, so it becomes better for you. Okay. Fine. So we have seen C3 plants and C3 pathway, which is nothing but Calvin cycle. We saw C4 plants or C4 pathway. Now there is another pathway as well, which is called crassulacean acid metabolism. It occurs in xerophytic cacti. Okay, xero means dry, extremely dry, and phyte, PHYTE, means plant. So the plants which live in extremely dry conditions are called xerophytes. So xerophytic cacti is plural of cactus, okay, as well as in succulents. So as you can see, here are the cactus, okay, these are all the cactus plants, and here are all the succulent plants. Succulent plants, you can include even aloe vera, are the plants where the leaf is very thick and it has some sort of jelly-like substance filled in between, okay, the leaves are very thick. So it is called succulent. The stomata on these plants open at night. Fine. Here the stomata they open at night. Examples are agave. Agave is very much similar to aloe vera. Okay, it looks the same, but if you know that tequila is made out of agave, then agave syrup, which is a sweet liquid like honey, okay, that is also made out of agave. So agave is used in a lot of things. So that is one type of succulent. It is one example. Aloe means aloe vera is one example. Pineapple is another example. Opuntia is your normal cactus that you see. That also. Then bryophyllum. Remember, bryophyllum is the plant where vegetative propagation is seen on the leaves. Fine. So bryophyllum, all of these are examples of crassulacean acid metabolism pathway plants, CAM plants. Here, the enzymes for both C3 and C4 pathway, whatever the enzymes we had seen in C3 and C4 are present inside the mesophyll cell itself. Okay, so there is no crans anatomy in CAM plants as well. There is just one cell which is mesophyll cell, okay, one type of cell, mesophyll cell, and in these mesophyll cell you have the enzymes which are required for both C3 as well as C4 pathway. At the daytime, here, let us now focus on the daytime. The pathway, C3 pathway, is active. Okay, so what will happen? This Rubisco, there is carbon dioxide. It will be used up by Rubisco enzyme. It will fuse this carbon dioxide with RUBP, convert it into 3-phosphoglycerate, and the Calvin cycle will start. When the Calvin cycle is going on, of course, it produces a lot of glucose. That glucose will be used to produce starch. Clear? At night time, however, C4 pathway is active. So at night, here the stomata are open. When the stomata are open, the carbon dioxide will enter inside. Okay. In the morning, you can see the stomata are closed. At the night, the stomata are open. So at night, when the carbon dioxide enters the mesophyll cell, this carbon dioxide is um, acted upon by PEPC, which is nothing but PEP carboxylase. Okay, phosphoenol pyruvate carboxylase. Phosphoenol pyruvate carboxylase will add this carbon dioxide to PEP and give you oxaloacetate. This oxaloacetate gets converted into malate, just like your C4 plants. Okay, just how we had seen in the C4 plants. This malate, what you are seeing here is your vacuole. So they get accumulated in the vacuole. Fine. At this point of time, there is no Calvin cycle occurring. Fine. Okay. So here you can see in the morning, when Calvin cycle is taking place, what will happen? The malate, which was stored in the vacuole, it will leave the vacuole. That malate will be converted into pyruvate and it will release the carbon dioxide. So can you see it is a bit of both? Okay, you can see C3 C3 pathway as well as C4 pathway. Clear? Here also same as C4 pathway. One glucose requires 30 ATP and 12 NADPH. Okay, it is same as C4 plants. Okay, everything is same as C4 plants, except here you do not have bundle sheath cells and mesophyll cell. You just have one mesophyll cells. So let us look at the comparison. Fine, this is the final comparison between C3, C4 and CAM plants. In C3 plants, you just have carbon dioxide, the stomata will be open and that carbon dioxide will diffuse. It will enter into this mesophyll cell. This bigger cell is the mesophyll cell. Okay, so it will enter this mesophyll cell. Okay, this is leaf, sorry, and this here is the mesophyll cell. It will enter the mesophyll cell. The first enzyme is Rubisco. It will convert carbon dioxide into 3-phosphoglycerate and the whole pathway will run. This glyceraldehyde 3-phosphate will then make glucose and later on sucrose. In C4 plants, you have carbon dioxide which will enter the leaf, and after entering it can enter the mesophyll cells. Mesophyll cells have PEP carboxylase which convert carbon dioxide into oxaloacetate and oxaloacetate into malate. This malate will enter the bundle sheath cell. It will release carbon dioxide. This carbon dioxide is then used for Calvin cycle and production of sucrose. Fine. So here what you are able to see is there is temporal, not temporal, spatial separation. Okay, there is a separate space where carbon dioxide will be accepted. Okay, this space which does not have Rubisco, so there is no chance of oxygen getting um, um, assimilated or fixed. Okay, there is only carbon dioxide which will come here. It will form malic acid and enter the um, bundle sheath cell, and in the bundle sheath cell, which is a very protected environment, you will find Calvin cycle going on. Clear? Now CAM plants, they do not have different kinds of cell. They have what is called temporal separation. Temporal means time. Okay, wherever you hear the word temporal, it means it has to do something with time. So here the pathways, they are separated by time. At night, when the stomata are open, what will happen? Carbon dioxide will enter. It will get converted into oxaloacetate, from oxaloacetate malate, and this malate will accumulate inside the cell that we saw, right, in the vacuole. In the daytime, what will happen is this malate, it will get released from the vacuole. It will get converted into pyruvate and release the carbon dioxide. This carbon dioxide during the daytime when the stomata are closed will be used to used for generation of sugar, okay, through Calvin cycle. So this is the difference, okay, this is the comparison. C3 plants, C4 plants where there is spatial isolation, and CAM plants where there is temporal isolation, okay, time, night and day. The cell is the same. This is one and only mesophyll cell. Right. After that, let us move on to the next topic, which is photorespiration. A simple observation is or was that more amount of carbon dioxide is produced in the leaves of C3 plants during the day. Okay, they took the leaves and they did some work and found out how much carbon dioxide is released by the plants. Okay, not accepted by the plants but produced by the leaves. Okay, in the morning, in the daytime. So they found that C3 plants, instead of using more carbon dioxide, they were making more carbon dioxide. Okay, so what was happening? What was the reason? It was found out that the Calvin cycle in the leaves of C3 plants, where oxygen was utilized. Okay, so instead of carbon dioxide, oxygen was getting fixed. So oxygen was utilized in presence of light, and carbon dioxide was produced. So the product, the substrate was oxygen, and the product was carbon dioxide. So this whole process is known as photorespiration. It is a futile cycle. Futile meaning you do not get energy out of it. Okay, it is just a uh, trash cycle. Okay, you are not getting anything out of it. You are just trying to regenerate your RUBP. You will understand when we study. Right. So the active site of Rubisco, this here is a Rubisco enzyme. It can bind to carbon dioxide as well as oxygen, and the binding is competitive. So carbon dioxide and oxygen will compete to bind with this Rubisco enzyme. It has much greater affinity for carbon dioxide when the ratio of carbon dioxide to oxygen is 1:1. Okay, when carbon dioxide is either more or is equal to oxygen, the amount of oxygen, it has greater affinity for carbon dioxide. Clear? But what happens in the atmosphere, you already know, amount of carbon dioxide is 0.4, while the amount of oxygen is 21%. Right. So what happens? Here is your Rubisco enzyme. This is your RUBP, which is a 5 carbon compound. The carbon dioxide, okay, when there is lower temperature and high carbon dioxide to oxygen ratio, this carbon dioxide will be fixed through Calvin cycle. Okay, you will get sugar and everything. But when there is high temperature and there is low carbon dioxide or high oxygen, in such cases Rubisco will use oxygen. It will bind to the RUBP, and this is called photorespiration. Clear? In case of carbon dioxide, the products are 2 molecules of 3-phosphoglycerate. While in case of oxygenase, part of the enzyme, okay, when it uses dioxygen, which is O2. So in case of O2, if it gets added to RUBP, then there is no carbon which is getting added. So RUBP, which has 5 carbon, will remain 5 carbon. When that 5 carbon compound it splits, it will give you 1 molecule of 3-phosphoglycerate and 1 molecule of 2-phospho. Where did it go? Okay, it will give you 2-phosphoglycolate. Clear? So this is how there is a difference between the carboxylase and oxygenase, the two parts of the same enzyme. Okay, and what is causing the phosphorespiration. Now, what are the conditions that favor photorespiration? Okay, if it is a futile cycle, if it is a trash cycle, nonsense cycle, what are the conditions that is causing the plants to undergo this cycle? So the first thing is high light intensity. When there is very high intensity light, okay, then it can cause photorespiration. Higher temperatures. If the temperature rises above 35 degrees or 30 degrees even for C3 plants, it causes a lot of photorespiration. When there is less carbon dioxide outside and more oxygen, okay, in this cases also there will be chances of photorespiration. The inhibitory effect on photosynthesis due to high oxygen. Okay, if there is higher oxygen, it will not allow photosynthesis to happen. Why? Because high oxygen meaning it will bind to the Rubisco and it will not allow production of sugar. So your photosynthesis is not happening. So inhibitory effect on photosynthesis because of high oxygen is known as Warburg effect. Clear? Right, now let us see here. This is the about the light intensity. On the x-axis you have light intensity. It is given in the units lux, if you know. Okay, and on the y-axis you have rate of photosynthesis. As you can see, at the lower light intensity, once when you start, there is light compensation point, meaning the minimum amount of light intensity required for photosynthesis. There is light limitation here, you are not getting enough light. As you increase the light intensity, okay, as you make the light brighter, the rate of photosynthesis increases. After a point, increase in light intensity will have no effect. Okay, so even if you are increasing the light intensity, the rate of photosynthesis has become stable. So this is called saturation. Okay, light saturation. And later on, if you still keep on increasing the light intensity, what will happen is your chlorophyll denatures. Okay, your chlorophyll molecule, instead of its original structure, it will become denatured. Think of this denaturation as when you put popcorn inside microwave or inside cooker, and when you heat it, it pops, right? It turns into popcorn. That kernel of corn turns into popcorn. So that is a sort of denaturation. Just think of that denaturation happening to the chlorophyll. So that chlorophyll bleaching or chlorophyll denaturation happens at higher light intensity. If the chlorophyll itself is not working, then of course your photosynthesis will not occur. So there will be a decrease in the rate of photosynthesis. Fine. This can also lead to photorespiration. Okay, so these are the conditions which favor photorespiration. Now let us see what kind of photorespiration the pathway is. In C3 plants, O2 binds to rubisco, thus the efficiency of carbon fixation is decreased. We will see again when you recall C3, C4 and CAM plants, you can recall that C3 plants are the ones whose mesophyll cells are open to all sorts of gases. Okay, it can be carbon dioxide, it can be oxygen. So photorespiration is seen more in the C3 plants. Rubisco, when it binds to the oxygen, it forms one molecule of phosphoglycerate and one molecule of phosphoglycolate, fine, which is two carbon compound. Let us see it here. You have oxygen. Oxygen will give you, when it binds to the ribulose 1,5-bisphosphate, it gives you one molecule of phosphoglycolate. Okay, the other molecule, which is phosphoglycerate, can go ahead and do the Calvin cycle. Fine. Now this phosphoglycolate, which is a two carbon compound, it will be the phosphate group will be removed in the chloroplast, and this will be transferred to peroxisome. Okay, peroxisome is another kind of organelle which has all types of peroxidases. Okay, it basically has all the enzymes which act against the reactive oxygen species. Okay, ROS. So now this phosphoglycolate, after releasing phosphate, becomes just glycolate. This glycolate is acted upon by oxygen. Okay, and it forms H2O2, which is nothing but hydrogen peroxide. During this process, glycolate is converted into glyoxylate. This glyoxylate finally forms the amino acid glycine. This much is the part of the pathway in peroxisome. This glycine will now enter the mitochondria. Inside the mitochondria, this glycine will release one carbon dioxide, so there will be decarboxylation. Okay, if you remove carbon dioxide from glycine, which will come of course from the carboxylic acid group, okay, of this amino acid. So that carboxylic acid group is removed, then NH3 is also removed, so the amino group is also removed. And there are multiple steps which will finally convert this glycine into serine, which is another type of amino acid, right? These steps are again not very much important, but just in case if any question is asked, or if you have somewhat knowledge about this, it will be helpful, right? That is why I'm going into a little bit of detail. Serine will again enter the peroxisome. Here it will get converted into glycerate, which is again a 3-carbon compound, and this glycerate will be converted into 3-phosphoglycerate using one ATP. So if you see, during this whole process, you are actually losing carbon dioxide. Okay, you are releasing one carbon dioxide. Fine. and also you are using ATP to synthesize this 3-phosphoglycerate. So you are losing energy, you are losing carbon dioxide during this whole process. Hence it is a futile cycle. Just remember this much. Here the oxygen, it binds to the RUBP, and with help of Rubisco it gets converted into one molecule of 3-phosphoglycerate and one molecule of 2-phosphoglycolate. The pathway includes many intermediates and reactions catalyzed by 3 organelles. Remember this also: chloroplast, peroxisome and mitochondria. There is neither synthesis of sugars, because of course you know that for synthesizing sugar you require carbon dioxide to be fixed. Now if there is no carbon dioxide added, how will you fix carbon dioxide, right? And if you cannot fix carbon dioxide, no sugar. So there is neither synthesis of sugar nor synthesis of ATP. In fact, ATP here is utilized for converting this into 3-phosphoglycerate. So this here is a photorespiration pathway. If oxygen binds Rubisco, it will convert it into 2-phosphoglycolate, glycine, serine, glycerate, 3-PG and RUBP. Okay, and then the pathway continues. On the other hand, this here is your Calvin cycle. If carbon dioxide binds, you get 2 molecules of 3-phosphoglycerate, triose phosphate, ribulose 5-phosphate and ribulose 1,5-bisphosphate. Okay, and the pathway continues. In C4 plants, the photorespiration does not occur, okay, or it occurs in very less amount, only sometime. Otherwise, in majority of time, it does not occur at all. Why? Because they increase the intracellular carbon dioxide concentration, okay. So in this bundle sheath cell where the Calvin cycle is taking place, there is so much amount of carbon dioxide more compared to oxygen that only carbon dioxide will be fixed. No oxygen will be fixed. Because of Cran's anatomy, the bundle sheath cell, they never come in contact with oxygen. Okay, only the mesophyll cell can come in contact with different gases. The bundle sheath cells, they do not really come in contact with oxygen at all. Okay. Third thing, mesophyll cells which are in contact with oxygen or which can come in contact with oxygen, they do not have Rubisco. So there is no point of photorespiration. Thus, C4 pathway is more productive. It uses more uh, carbon dioxide. Okay, it may be using a little more ATP compared to C3, but it is more efficient. It is giving you more amount of glucose, and also it can tolerate higher temperature. So C4 is actually a good evolution. Okay, it is helping the plants conserve energy from photorespiration. Right, so this is the table which was given in your textbook. I thought I would just mention it here. There were different questions which have been asked, and for C3 plant and C4 plants, you had to fill up all these gaps. So I'm not going to go through all of these. The reason is we have already discussed at length during this lecture. So you can just pause here, and if you want to fill up your textbook or if you want to just refer to it, you are welcome. Okay, see you next time.